the one with the swivel chairs, if you know which class. It's just right down the hall. It's pretty fun. Okay. Thanks, Sean.
Okay, 2 p.m. today. I was up from 3 to 5 last night. So, yes.
So we just call it a day and go.
Pull out your clickers. Let's do a little. Let's see how well we're understanding what we've been doing. First question. Oops. Participation. But questions like this will be on the test. So, okay, that's it. Ready? No, we'll stop it. Okay, so that's correct. C is correct. We've never talked about enhancer genes, and that was just a red herring. Seconds. Okay, you guys definitely have that one down, don't you? Okay, 5 seconds. All right, let's see if you guys can convince each other that you're right. Okay? And then revote when you've done it. Yeah. That's what the precondition is. The predisposition is back to both. Because we all say we all have the statement is true. If it said a predisposition to cancer, you could have one allele of tumor suppressor stability. I would say true. But if it says that you must have one, yeah, it's not, you can have a predisposition. Because don't read too much into it, Jordan.
I'm not saying you're wrong, it's just really confusing that you're saying.
All right. Go ahead and revote. I'm missing about 6 people.
Yeah, okay.
All right. Okay, so from what we've learned so far, why is that true? I heard Jordan had a great point, but he's reading too much into it. I'm not saying you absolutely have to have these to be predisposed but from what we've talked about in class, this is what we've talked about, meaning this is what we've said is predisposed. Yeah, Michaela?
Can you have it on
Great question. I was hoping someone would ask. If you have it on if you have a mutation in an oncogene, why would that not fit into this? Jamie? Yeah, in an oncogene, remember, okay, here's a chromosome with an oncogene on it. Chromosome from mom, chromosome from dad. Here's the oncogene, let's call it RAS. If you have one mutation, if this guy's overactive, then that's a stuck accelerator. So you don't need both of them to be mutated to be a stuck accelerator. Does that make sense? It wouldn't be so much a predisposed to cancer as you probably would get a tumor. Yeah, if it's in the right kind of tissue. Great. Is that what you were going to say, Nicole?
Yeah.
Great. So, if a person inherit, like Jane and Sam, if they inherit from their dad an APC that's mutated, APC happens to be a tumor suppressor gene. This normal copy that they have, oh sorry, that's from mom, isn't it? So this normal allele that they have from mom will be able to regulate those checkpoints normally. It will be enough to regulate those checkpoints. But, unlike most people who have two normal ones, why do they get cancer more often? Here's Sam, this is Sam, right? Why is Sam more likely to get cancer than a normal person? The normal person still has the APC gene. Yeah, he's already coming into life with one hit. So all he needs is a second hit. So here's what's happening with you guys, is that throughout life, you're probably getting one hit in a cell, randomly. If that happened to Sam, he'd get cancer, but with you, you still got a normal allele. So the chances of him getting a hit and the chances of you getting a hit are the same, but if he gets a hit, he's got tumor. If you get a hit, you're still going to be normal. Does that make sense? Okay? So, that's why it's true, is that he's predisposed, he's already got one hit in that tumor suppressor or stability gene. So those are the two gene types, tumor suppressor and stability, where you need two hits. With the oncogene, you just need one hit. Oh, that's it. Okay. All right. I wanted to revisit this really quickly because we had to go over it quick. We had to do it fast on Wednesday, but remember, this is Sam. And this is his open reading frame, and we didn't really get to talk about what an open reading frame is, but what did you guys get from your reading and from this activity? What is this blue line? When we say this blue line is an open reading frame, what are we really saying? Yeah, it's a string of code. Yeah, so the reading frame, remember, DNA is read in triplets, right? 3 bases at a time. So it's read in triplets. But when we talk about a reading frame, the question is, do we start reading it here, here, or here? If we start reading it here, then it would be 1, 2, 3, 1, 2, 3, 1, 2, 3. If we start reading it here, it would be 1, 2, 3, 1, 2, 3, 1, 2, 3, right? If we start reading it here, it would be 1, 2, 3, 1, 2, 3. So there are 3 possible ways you could read the DNA. But the only one that gives you a full blue line, there's only one way that will give you a full blue line. So this means when you see blue like this all the way through, that means there's no stop codons. So this was the first time right here where we had a stop codon. So all of this was the complete APC sequence. And what did we see with Sam? He had a stop codon right there. And so basically what happens when this mRNA is translated is that the ribosome reads mRNA and then it gets here and it stops. And it makes a protein that big. And then it starts again, and it makes this. But this, by itself, will not give you a normal protein, neither will this by itself. So he basically has a non-functional APC. Now, my question to you is, what if, instead of having this mutation, Sam had some other mutation somewhere, and when you went and did the reading frames, you saw that Sam had a normal blue bar all the way across, but he still had colon cancer. What would you think is going on there? Do you understand my question? So there weren't any stop code. The mutation didn't create any stop codons. It was a normal length protein, but he still had a mutation. He still had colon cancer. What do you think's going on?
So you're saying that it didn't stop?
It didn't stop until the normal place where it normally stops. What do you think happened, Nicole?
It was just, instead of it being a nonsense, right, it's right there, it was a nonsense mutation.
Oh, okay. And what does that mean?
Uh,
How could he get cancer from that?
It's like a change in the amino acid, so it was the wrong amino acid.
And so how could he get cancer from that?
So,
That's great, Nicole. You got us a long ways. Jared?
So instead of having the stop codon being chopped in half, it would be a different shape because of the different amino acid in the middle.
Yeah, so instead of being shorter than normal, what that mutation might do is it's still the normal length, but maybe that mutation caused that change in amino acid caused it to be a different shape, like Jared's saying. And so it still can't do its job, even though it's the normal size. That's possible. That happens a lot. Remember, we've talked about how important shape is to proteins, and their shape depends on the amino acids they have. Remember sickle cell anemia? That was a great example of one change that completely changed the shape of the protein. Great, good job, Nicole and Jared. So I wanted to cover that. And, okay. Okay, that's all I wanted to do. We won't do part 4. It's not really the meat of what I wanted to cover for that unit, so we won't do that. You can read about it if you'd like. I did want to come back to something from our first case study, though.
Oh, yeah.
We never finished. Okay? So, but before we go here, are there any questions about APC in that case study? Before we move on. Any question about tumor suppressors, oncogenes, stability genes? Do you guys think about this question? Why don't you tell your table mates, why does chemotherapy make us so sick?
Oh my gosh.
You guys have it now? There's actually more. It does. That's the answer. Spread around. Chemotherapy with restoration. You would die. Some people choose to rather. Some people choose to die rather than Okay. So what are your thoughts?
For my job. He
What's going on with chemotherapy that's making us so sick? Yeah, Kimberly?
My knowledge of chemotherapy is kind of limited, but isn't it so that chemotherapy goes to kill cancer, the cancer cells, but isn't it possible for them to affect the regular cells as well?
And how does it affect those regular cells? That's exactly right, Kimberly. How does it affect those regular cells? What's that? Who said divide? I heard the word divide. Bryce?
Doesn't allow them to divide.
That's one major thing that it does. Jared, did you have something else?
I was going to say, it specifically targets cells that replicate quickly.
Yeah, remember we talked about Taxol? This is one of those, why do we use chemotherapy drugs? Remember what it does? Here we have our cell about to divide, and you create this skeletal structure in the cell that grabs both of those chromosomes and pulls them apart. These are like, you can think of these as skeletal tubule structures, right? They're called microtubules. But Taxol breaks that up so the cell can't pull the chromosomes apart, and it aborts division. And this is important, Jared, what Jared said about it. So Taxol will work on any cell that divides, but what are the cells that are going to hit hardest? The ones that divide a lot, they're really going to struggle because that's their bread and butter, is to divide a lot. And what kind of cells do that in our body? Skin cells, blood, good. The lining of our stomach, I mean, we're basically eating our stomach with acid a little bit every day, right? We've got to replace that tissue. Mike, were you going to?
So isn't the theory that you can kill off more of the bad cells than the good cells?
Right, okay, so that's the hope then, right? Is that we're willing to sacrifice some of our cells that are dividing quickly because we know that they might eventually recover, but we're hoping that it will be enough to stop or slow the cancer cells. That's why people's hair falls out too, right? Because they're not producing, those cells divide rapidly too. Okay, great. So that's why you get so sick, is because you're really attacking your own healthy cells, and that's why Lauren was asking, how long do you have to do chemotherapy for? Well, usually they do it in rounds, like you have your first and second and third round of chemotherapy with rest in between. Because if you gave the person chemotherapy continuously to kill your tumor until your tumor was dead, you would probably die before your tumor would. So what they hope to do is kill some of the tumor, give you time to recover. Hopefully the tumor doesn't recover also, and then give you a second round of chemotherapy and give you time to recover. It's really nasty. It's a terrible option, but kind of what we have right now. Isaac?
How is Taxol given to you? Is it like just like rubbed on your tumor spot, or?
Good question. No, usually you have an IV, and it's put into the IV so it goes straight into your blood. There are some chemotherapy drugs that are orally given now in pill form, but I think traditionally, for the most part, they're given straight into the blood. And so what happens, that's why it would be nice, wouldn't it, if we could find a drug that just goes to the tumor and leaves your stomach and your skin and everything else alone. And believe me, there's plenty of people who are looking at options for that. And so they're studying tumors really closely and saying, what is it different about these cells compared to my stomach cells? And can I make something that will recognize and target those tumor cells and leave everything else alone? That would be a great way, right? That would be a great way to kill your cancer.
Why is it that when people lose their hair and they have chemotherapy, sometimes the hair doesn't?
You might be killing off some of the melanin-producing cells. I would assume that most of the time it goes lighter rather than darker. But
Well, my friend, she lost her hair and it's blonde and pink.
are you sure she wasn't just dyeing it?
That is a good question.
I don't know. I can think of a way that it would, I can imagine it coming back blonder, but I have a harder time imagining how it could come back darker. Because if you kill off some of the melanin-producing cells, I could imagine chemotherapy killing them off, and so you'd have lighter hair. But okay. Okay, angiogenesis. We haven't talked about this, but I think this question will help basically teach you about it. So read through that and answer it best you can. Okay, 10 more seconds. Okay. you guys discuss this one. And revote when you come to consensus, revote. Okay, go ahead and revote. Sometimes there are good, better, and best answers, right? I think this question probably fits that bill. Okay, ready? Oh my gosh, that's pretty much identical. Let's see. Yeah, just people voting overall. All right. Why do you think, who answered B and would like to argue why that's the best answer, and then D, why is that the best answer? Okay, we've heard from Jared and Kimberly lots. Anybody else? Cody?
I'm not sure what metastasizing means.
Metastasizing?
But, yeah,
So you answered D?
I answered B.
You answered D because you didn't know what a word meant.
It sounded like a big word, but I figured, you know, it could be the reason why it sounds good.
Okay. Christina?
Just use a lot of context clues. I think B because one of the questions we're not talking about metastasizing, it's talking about tumor growth, and that one fit best to me. And when we were talking about it, we were talking about EGF and tumor growth and how it fit best with cancer.
Okay. Good. Anybody want to talk about D? Bryce?
I mean, I know what metastasizing is. It's just where the cell of a tumor breaks off and travels through the bloodstream and goes to another part of the system.
Let me show you a picture of it,
exactly.
you guys, Cody and everyone else. So, yeah, describe what's happening here, Bryce.
It breaks the lining of
What does?
the tumor.
Tumor, okay.
It breaks the lining of the blood vessel, of the vein.
Okay.
And it travels through and crawls around until it reaches another location, breaks through again, and starts to grow into a tumor.
Yeah.
And multiply.
That's metastasis. Does that make sense, Cody?
Yeah.
That process?
So D was right.
Well,
but with back to the question, I chose B just because on the factor with the question it was talking about supply of nutrients in the tumor with angiogenesis, and I mean a cell moving around could be the cause of that, but to me it sounds more like the supply of, I mean, how a cell multiplies by having nutrients and being able to grow.
okay. Michaela, what were you you can add something else?
I was just gonna say, like, both of them seem right, but for D, like, if there's more blood cells, or, yeah, blood vessels around it, it's gonna have a higher chance to get into this and move around.
That's a very good point. That's a very good point, Kimberly.
I said B because I felt that kind of led to D. Because not metastasizing, since that's the process of just breaking through a vessel and forming new tumor somewhere else, then in order for that to happen, this tumor has to grow. And since it says that it needs new blood vessels to grow, then it made sense to me that the drug that binds to EGF would stop it from growing, which in turn would stop it from growing.
Yeah, that's a very, I like that explanation. Kind of this D could be an answer, but B would have to happen first. It would have to grow first before it could. Okay, so why not A? Why didn't anybody pick A, did he?
Because we weren't talking about what side effects the angiogenesis process would.
And what would you expect? Something that stops cell division versus something that stops new blood vessel formation. Which do you think is going to make you more sick? Well, how often do you think you form new blood vessels in your body? That doesn't really happen that often, right? I mean, once you've got your circulatory system set up, you're not really forming a lot of new blood vessels. You might bruise yourself and you might have to reform and heal blood vessels, but we're not sending blood to places that have never had blood before very often. So cell division is happening a lot, though. So something that's disrupting cell division is probably going to disrupt your health more than new blood vessels. And C, why wasn't C right? Why didn't any of you pick C? Carolyn?
In the blip before the question, it says that the tumors secrete higher levels of VEGF.
Yeah. Yeah, higher levels. So we know that, of course, if we need to form blood vessels, we need to have that VEGF around. So it's not like we can't. It's not like that's never around. It's just that these tumor cells are particularly adept at making a lot of it. Okay, so I would agree with you guys that B and D both could be good answers. I would probably put B above D, just because you have new blood vessels doesn't necessarily mean that tumor's going to find its way easier into it. It doesn't necessarily mean, although I really like Michaela's argument, it probably increases the chances. But what it definitely could do would stop them from growing. It could definitely do that. So, good. Cancer detection and treatment. Won't go into this very much other than to just state the obvious. The earlier you catch it, the better. So these are some of the things that you might be concerned about if it happens, if it lasts for a long time. But don't worry about knowing all of these things. It's a caution there. Okay, Cody, this question's for you and everybody else. But Cody definitely has to get this one right. So. Okay, 5 seconds. Okay, that's true. So the problem with metastasis is now, yeah, you could go to that original source and remove it, but now you've got it spread to other regions, so you've got big problems. All right. now allow, oh. Happy ending, Abby's ovarian cancer is in remission for 10 years. She graduated from college. She's living happily with her husband Charles and her 4-year-old adopted daughter. I don't know if that's true or not, or if that's completely made up. I actually don't know. So, what's that?
That's why it's
That's why she's an adopted daughter. Yeah. Okay. So we have one more thing to go over with cancer. Okay. Let's go through. Let's go down here. So what kind of things cause cancer? We've talked about this quite a bit, but I want to mention a couple things to you. If you guys have never subscribed, if you've never listened to this program, I highly, highly encourage you. I think this was one of the links I gave you at the beginning of the year. Radio Lab is perhaps the best podcast if you're into podcasts that's around. It is absolutely fantastic. And there's one podcast called Double Blasted. Definitely listen to that. It's what are the 2 cities in Japan that were bombed with an A bomb in World War II? Nagasaki. This podcast episode tells the story of a man who was working at Hiroshima the day the bomb went off and made his way home to Nagasaki for the next day's bomb. So he got bombed twice and had incredible amounts of radiation pass through his body, and tells the story of him and his life afterwards. When we talk about atom bombs going off or X-rays, really powerful rays that can pass through your body, they're often called ionizing radiation. I won't get into it very much other than to say it can cause breaks in your DNA. Yeah?
So it's just everyone says about the microwave?
I think that's a myth.
Or the cell phones in your pocket?
That is, and that's up for grabs still. Yeah. Yeah. They have done it. They have done a study where they found that people are getting brain tumors on the side of the head that they hold their phone to. So, but it's really, the verdict's out. There's not enough information yet to say.
So switch off.
Yeah. So you get it on both sides. Yeah. So that would be non-ionizing radiation. Another common form of non-ionizing radiation is UV light from the sun. So these are your X-rays and your nuclear rays, and these are basically other rays. Both of them can cause mutations, but these are much more damaging to DNA than these are. So chemicals, it's not a surprise to you that certain chemicals can cause mutations in your DNA. Certain viruses and bacteria can also. Didn't you read about viruses that can insert into your DNA? Did we talk about that? And disrupt genes. So if a virus has genetic material too, and the way it does its job is it inserts its genetic material into yours. Well, what if it inserts its genetic material right into the middle of a tumor suppressor gene? Then, it could create a problem for you because you don't have that tumor suppressor gene anymore. Genes, we've talked a lot about that. Lifestyle, everyone knows that smoking can cause cancer. And getting old, because you accumulate mutations over time. Okay?
I'm going to be like Peter Pan.
Yeah.
Just keep saying that.
Okay. These are all the chemicals that are in cigarette smoke.
Wow. Smoking, we're too now.
So how would you know? let's say you've identified all these compounds that you find in cigarette smoke. How would you know which one of these causes cancer?
Start.
Human experimentation.
Start bringing each one together.
What's that?
Take a mouse and introduce each chemical to them.
Exactly.
That's where cancer begins.
Exactly. So here's something you need to think about. You'll often read in the news that such and such chemical causes cancer in mice. Now, should we just panic and run for the hills when we hear that?
No, because
Mice are different, and often what we find that some things work in mice and not in humans, and the other issue is that amounts matter. So how do you know, so it caused cancer in a mouse, how do you know that you didn't give it 5 million times the amount that a human would normally get? So how do you know amounts matter, and so often when we make tumors in mice, that amount would not make a tumor in a human. And so don't get all panicky when you hear that a certain thing like Coca-Cola makes tumors in mice. They gave that, is that the one that we talk about, that you hear about, or what's the one that, I know there's some common, yeah, like the aspartame, like the artificial sweeteners, right, that they'll cause cancer in mice. Well, guess what, they gave those mice incredible amounts of those artificial sweeteners. Way more than you would ever drink in a lifetime, even if you're Brother Coleman, who always has the back over. So, just be aware of that. So viruses can create mutations in your DNA. So what happens is that a virus has its own DNA, and if it inserts that DNA into your cell and disrupts a gene, and that gene happens to be involved in cell growth, then you may get altered cell growth. Not because the virus is making you grow, not because the virus is making that cell grow irregularly, but because it broke up a gene that's involved in cell growth. Okay, there are various viruses. I don't want you to worry about the names of all these. You've heard of HPV though. There's a vaccine on the market for this right now. It causes cervical cancer. The FDA is recommending that any girl younger than or older than like 13 get this vaccine because they assume that most girls at 13 become sexually active, and so this is passed. It's a sexually transmitted virus, and it can cause cervical cancer, and it can make you at risk for other types too. HIV is a virus that causes AIDS, but it also increases the risk of you getting something called Kaposi sarcoma, which is a kind of cancer. And herpes virus also increases your risk for Kaposi sarcoma, and let me show you how that works. So, what HIV does is when it affects you, it lowers your immune system. When your so it basically weakens your immune system, and that once your immune system's weak, then other viruses that normally your body could fight off, your body can't, and one of those viruses can cause cancer, this Kaposi sarcoma. So you weakened your immune system, you can't fight off that virus, and that virus inserts into your DNA and gives you cancer. Let's not worry about these, other than to say there are viruses that can cause cancer. H. pylori is a bacteria. This isn't a virus, but it's an actual bacteria that can cause stomach cancer. And most of the world has this in their stomach. All right. We've talked about heredity. Here's some things that you shouldn't do. Okay. Don't pack up yet because we still have 5 minutes, and I'm skipping through this because I want to get to to this part, okay? So, pull out a piece of paper and do this.
Okay, how do we do this quickly? Forget that.
Yes, these will be on the test. Okay, so that's more or less it. Now my question to you is, which one of these has the most? Carcinomas do, right? Carcinomas are cancers of epithelial tissue. What's epithelial tissue?
Outer.
Epithelial tissue, more or less, it's not the best definition, but it'll work for this class, is tissue lining cavities, like the tissue that lines the inside of our stomach or an intestine, or tissue exposed to the external environment, like our skin, or inside of our mouth, or breast tissue, for example, is epithelial because some of that breast tissue lines the cavities of the milk ducts. Our eye, our colon, are exposed to the external environment. Kidney cells line ducts. They line the kidney ducts that urine flows through. lungs are exposed to the external environment. Pancreas, line of gland. Prostate, also a gland. Skin's exposed to the external environment. Why are so many cancers? In fact, 80% of cancers are thought to be carcinomas. Why? They're exposed to the environment, and they're often, so they're exposed to the environment, and they're also often dividing quite often. Because they're exposed to the environment, but also because they often are involved in glandular processes, so they're releasing things. So we find lots of cancers or carcinomas. Now, which are the easiest cancers to remove? Carcinomas are the easiest to remove too, usually because they're exposed. So I wanted you to understand those two. So you need to know what these different kinds of cancers are and the things I just taught you. So good luck on your test, and we'll see you Monday.